First of all, welcome to today's podcast. Let's start with Swapnil, sir. You have extensively worked in the field of virology, especially in the hepatitis E virus replication. Could you give us an overview of your research work that you have carried out? And I would love to know your current status of work.
Okay. So, thank you for having me and best luck and congratulations for your YouTube channel. I am founder and director of Paeon Laboratories, Kolhapur. So, currently we are dealing with academic research as well as small scale industries. So, we provide them kind of customized solutions as well as consultancy. Other than that, we provide training programs for BSc, MSc, and PhD students with related to DNA, RNA, and protein molecules, isolation, purifications, kind of, or the cloning, recombinant protein expressions. So in NIV, I was working on hepatitis E virus, and my main focus was on papain-like cysteine protease and its role in overall.
When it was happening?
It was in 2012. So I qualified CSIR NET after my MSc and then joined there as PhD or a junior research fellow.
How did you start your journey? Like, what led you to, choose your career in virology, particularly?
So it was like, I will start from my birthplace. So I was born and brought up in Kaulge, Kagal Taluka. After that, I did my schooling in Genome, Kagal, Kolhapur. So during Navodaya, I was a good student in academics as well as sports. I used to play cricket. And then also I was good in cultural activities. I used to direct and act in plays. So somehow lots of expectations were there from my teachers, my parents, and my relatives. But after 10th, I left the Navodaya Vidyalaya. Then I joined 11th, I joined in Vivekanand College, Kolhapur. But somehow due to my health issues and the burden of that expectations,
Family expected you to be good in the academics.
Not exactly. They directly didn't force me. But due to their expectations, I was considering
in academics.
Okay, in academics. So I was expecting I will be a doctor, a good doctor, but I couldn't. So somehow I went in depressions, and that traumatized me for a very long time. So around my degrees and my master's, almost 5-6 years, I was in depression. So I was afraid of exams. So what I did, I threw out the academic syllabus and I started following kind of philosophy, but philosophy related with the science.
Okay.
So other than syllabus, I used to read about how might be evolution has happened. So how there might be the origin of life, and what is that antim satya in so, and how to live an ideal life, that kind of things. And 2-3 kind of things struck me. So while studying kind of philosophy, I interact with religious scholars. I read religious books. Then also I read kind of articles which are related to the philosophy and other things. And somehow I thought in Bhagavad Gita there is a word like Sthit Prajna. So I thought I should be like that only. In every situation, my mind should be very much stable. And one more quote was there. I read. It is like go towards your origin, and you will actually understand the meaning of life. That kind of things. So I took it literally, and I started running in backward. So in the beginning, I wanted to become a doctor, which I didn't.
You worked in NIV. I mean, you were doing PhD, right? So instead of choosing R&D, like let's say research as your career, what led you to choose, to start a new startup instead of carrying professional journey?
Okay. So it will come again to the same point. So after that traumatization and depression, I started isolating myself from the people. So, and I started following like animals, plants, and bacteria. So I thought like I shouldn't concentrate on to the people or idolize the people. Rather than idolizing humans, I should idolize animals. And the reason behind it, like uh, humans can express, they have the languages, they can express themselves. So I thought uh, more than humans, animals are better. I thought like animals, they are less expressive as compared to the humans because they are not that much expressive like languages. So I started following them. So I started getting interest in the animals. So after few days or after few months, I thought no, animals will not be my idol. I should go a little bit back to the plants because throughout the life they stay at a single place. Still they are less expressive as compared to the animals, but still they are more stable than even animals. So I thought plants should be my idol. So I did degree in BSc botany. So then I entered for the masters in biotechnology. Then while studying again masters, I thought more than plants, bacteria should be my idol, or I should focus on them to get that stable mindset. Why? Because again, plants or bacteria are more stable. They don't have kind of specific system, organ system, or they don't have brain. So within the single cell, they are doing everything. And at the end of my masters, I thought more than the bacteria, viruses should be my idol because even they don't have the cells, they don't have any kind of metabolic pathways.
In which subject you did master?
In biotechnology. So I started following viruses, but I wasn't sure that I will do work in virology or virus field. And also, internet that was not that much accessible, so I couldn't find out a specific route to go to a national institute or anywhere. But somehow that remained subconsciously in my mind. And after MSc, I joined a lab in Pune, Shivaji Nagar, in agricultural campus. So there was a lab. It is newly lab established in 2000 around 9-10. So rather than using traditional methods, they want to find out the adulteration in the seeds by doing DNA isolation and PCR by using primers. But when we entered there wasn't any kind of technical staff. They didn't recruit. Also, the old staff was doing by their traditional methods. So we got that space, and actually, the research interest start from there. So we did a lot more troubleshooting while optimizing the plant DNA isolation, and after that, PCR optimizations, and that kind of generate interest in molecular biology. And the other part is I was very fond of Hollywood movies. So their actors will be like scientists or army's best kind of soldier. So I thought they should be my idols. So I started kind manipulating. One day I will have my own lab and I will work there day and night. So in the nights I will have that coffee mug in my hand and I will be intensively working in the nights. So that kind of dream remained inside me for a very long time. But after doing, after completing work from the NIV, so then I started to think about I should have my own lab. So somehow I found out I will not be good in kind of government agencies, and also I will not fit in private companies. So I chose my own path and I followed it.
Coming to viruses, I want to know about your perspective regarding viruses. So there has been a big debate since long whether viruses are living organisms or non-living organisms. Some people also claim that they are borderline between living and non-living organisms. Okay, so since viruses have to be dependent on host cell factories, let's say for in order to reproduce and in order to metabolize. So in this sense, I mean, if you consider whole scenario, people are divided in their opinions. Some people say that viruses are living organisms. Some people say that viruses are non-living organisms, and some people say they are borderline between them. So I want to know about your perspective. What, according to you, are viruses?
Okay. So actually, kind of I idolized viruses. So for me, they are kind of living. But in scientific fraternity, they don't consider viruses as a living. But for me, they are living. Why? Because hardly with 8 to 9 genes, they are fighting with almost 30,000 genes in case of human genome. And they know how to tackle those 30,000 genes, or once entered into the cell, how to uh, kind of combat with the defense system of the cell line. So in any cell line, kind of humans or animals or avians. So that's why I consider them as a living. And one more thing, like philosophically, why I follow them, they are like simple living and high thinking creations. They don't have much more to carry with themselves. So they totally they create their own world inside the cells. They work there and they exit. So this is the philosophy I try to follow. Don't try to create something, or don't create about the physical things. Just be as usual as you are and live your life. And also they are, while infecting to the cells, they didn't create more chaos. Actually, the chaos is from cell side. So they just want to go inside, replicate. So in case of HIV, you can see when it comes to from mother to child.
No, he is asking about whether should you consider, what's your perspective on considering virus as a living or non-living?
Okay. So it is just, for me, it is living only. So not more than that.
Yeah, means addition to him, like why even he or any of who knows the biology, like basics of the biology, so why it is basically considered like it is maybe non-living thing and maybe living thing. Now why it would be living thing, you already explained it. Can you like shed some light on other side, like why virus is also considered as a non-living particles?
Okay, so we defined, to avoid the chaos, we defined these are the living criteria and these are the non-living criteria. So as they don't have their own metabolic pathways, or they kind of don't have other transcription translational modes.
Okay.
So we generally scientists consider them as a non-living entity.
Or we can literally like synthesize them and as a chemical like any other chemical particles. So like that particles you can extract them, and without host they will remain as a normal particle, what we call virulence or viroids. What we say. So
they can remain in the atmosphere, but not for longer time.
Okay.
So they always need a kind of host to replicate them.
So can you shed light on how do viruses replicate? I mean, how this replication thing happens inside them?
Okay. So one more in addition to that, generally viruses have both the DNA and RNA as a genetic material. So throughout the evolution, organisms choose DNA as their genetic material, but viruses, they run separately. They run their timeline separately for the evolution. So they have DNA as a genetic material as well as RNA as a genetic material. So accordingly, DNA viruses, they will have their separate replication strategies, and for RNA viruses, they will have the separate strategies for the replications. So in case of hepatitis E virus, it is a positive sense RNA virus. So positive sense means once it enters into the cell, it will work as a messenger RNA, and it will direct start the translation rather than starting the replication.
Can you explain about messenger RNA? What exactly it is?
What has happened during the evolution, DNA has considered as a kind of storage material. Okay. So everything what has happened with during the evolution is stored into the DNA.
Information storage.
Information is stored throughout the evolution. So DNA is like data storage. So it won't don't want to work for anything. So what organisms choose the middle part? Let's take only required things. As cells are more economical, so they will invest the energy only when they want. Okay. So means like heart cells, they will behave like heart cells. They will not behave like brain cells. So they have decided their work. They have decided which proteins or which expression should be there. So accordingly, which gene they want to express, they will first convert into the messenger RNA, and then actual proteins will work inside the cell. Okay. So actual hard work will be done by the proteins only.
Okay.
I have a few questions for you. Which is your best movie you have ever watched?
So Iron Man was my best movie in the sense. No, there are many best movies from Christopher Nolan's also, but ideally I followed the Iron Man movie.
Who is your favorite actor?
Yeah, Robert Downey Jr. only.
We will move on to the hepatitis E virus replication. Before that, I would like to know from your side. See, you came from animals to virus. Okay, a complex organization to a simple organization, but again effective organization. For example, millions of genes are present in case of animal and plant cells, but still you choose to be part of virus, that is to study on the virus who are having thousands of genes. So cancelling billions of genes, you are working on thousands of genes. Again, specificity is there, considering the Christopher Nolan category, that I will use only those things which are required or which are necessary. Again, considering that into mind, the characters of virus, like it stays silent before infection, its performance once it infects, and technically it is very smart, considering the evolution point of view. Do you consider yourself as a virus?
I try to idolize them because
I think some of the characters that you are studying from the viruses are inherently or maybe fortunately because of you are working on that have been in your body currently that you are considering yourself.
Yes. It's always when you, as a human, when you want to follow the good life, you have to be more stable. It is like quantum computing, 0 and 1 simultaneously. But a little bit of imbalance will create the havoc. So viruses once enter into the cells, they try to soothe the cells so that defense mechanism will not harm them. But due to the kind of flight and fight modes, cell responds towards them as it as an invasion, and they start their defense system. Otherwise, they want to just remain inside the cell and multiply. See, we have the pandemic.
That's why I'm asking whether you learn from the virus or because you're a fan of virus, or maybe you're working on virus because your characters and virus characters are somewhere similar.
Yes, that is what I wanted to.
Because coming from plants or animals, then bacteria, and then virus. So it's a kind of journey that you had. So it's kind of you're choosing something to be more idolized that will be similar to you.
Okay. Yes.
And then you finally have virus where your characters are matching, and now you are like, yeah, this is the one where I wanted to work, or this is the one onto which I want to do work.
Yes.
Because it is quite similar to me.
Yes. That is what just I wanted to express this. So pandemic was there. So in the beginning, COVID, like SARS-2, it was more virulent, but now it is not that much virulent. So until the humans will be there, now SARS-CoV-2 will be there in the humans. But what it has happened, it has lowered down its virulence capacity. Okay, so mildness is the best to live as a human, to interact. Means what we think, suppose something is happened in front of you, immediately react to that thing. So viruses teaches you, don't react, just sit cool. When there is a need, just come and express yourself. Similar, again, I will tell you about the DNA viruses like Epstein-Barr viruses and herpes viruses. We have infection since our childhood, but they will show the expression in latter stage of our old age, because otherwise there will be 2-3 more criteria. If your immune system is weak or your immune system is compromised in case of cancer therapy while it is going on, or there is organ transplant is going on, then only they will come and express. Otherwise they will live inside you throughout your life.
Okay, let's start with the hepatitis E virus replication. As we have understood that, I personally have understood that virus is the kind of thing where you did the most of the work. So let's start with the infection, how hepatitis E virus impacts, and the cycle of replication and all other things.
So hepatitis E virus, it has the origin in India only, but later on by retrospective studies, we found out that in 1949 or during 1955, there were robust outbreaks of hepatitis E virus. Almost 29,000 people were infected by hepatitis E virus. So its transmission route is fecal-oral. So it comes through contaminated water resources by our food. So in case of developing countries, it is due to the kind of eating half-cooked meat. But in developing countries like subcontinent of India, subcontinent of Asia, like India, Pakistan, Sri Lanka, Bangladesh, we have waterborne outbreaks of hepatitis E virus, but first time reported in 1983. But generally what we do in virus field, we try to store the patient samples. So when we did the retrospective study, we found out that hepatitis E have the origin from India only. So it is around, documented is 1949. So before that also there might be the outbreaks for hepatitis E virus. So again I said like this, it is a RNA virus, it is a positive sense virus, and almost 2/3 of the world's population once in life it has been infected by this hepatitis E virus. But the plus point of it is self-limiting. Again, it is a mild. But if you are immune compromised or you have the addiction, or in case of pregnant women, it can be fatal up to 20-30%. So I started working on molecular base for the hepatitis virus. So generally about viruses, you can study in two different aspects. One as a molecular and one as a clinical patient sample based. So you can do the patient samples and you can follow the changes in their antibodies and other biomarkers expressions. So my work was related to its core. So hepatitis E virus, it has a 7.2 kb genome. So it is a RNA virus. So almost 7,200 nucleotides were there. So out of that, almost 5.1 kb is non-structural protein, means ORF1. And ORF, there are three open reading frames. So first one is ORF1, where non-structural proteins are prepared. And
What really is the name of ORF?
It is open reading frame, means actual protein synthesis will start in that frame. And ORF2 and 3 were like capsid, and 3 is like it will help in viral regression.
So these are like different sections of genome. Okay.
So first part is non-structural, means it doesn't have role in structures, but it will help you or help the virus to replicate inside the cells. So my study or my interest was in PCP. So it is non-structural protein. So it will help the virus inside the cell to replicate or to establish itself.
What is PCP?
PCP is papain-like cysteine protease, means it folds like papain enzyme from the papaya, but it has its own sequences.
What it does?
So it has role in polyprotein processing. So generally what happens in case of also one more interesting facts about the hepatitis E virus, it resembles with the plant virus. So there might be the chances somehow during the evolution it might have shifted from plants to the animals. And what generally happens in the plant viruses,
I would like to ask one question. Why this transition occurs from plant to animal or animal to humans?
every time virus will try to find out best host.
Why?
It is the kind of their evolution strategies. So to find the and which will be the available in abundance, it will choose. So
More than evolution, it will be like survival strategy.
Survival strategies also. So currently what we are facing the zoonotic transmission to the humans, and the big reason for it are also the deforestation. So we have vanished the natural habitats of the animals.
Okay.
So their viruses also now trying to find out the new host, which will be the humans, because they are abundance in currently in nature. They will be the abundance. So they choose always, and it is kind of evolution running parallel to the what actual evolution happening with the higher organisms. So there are viruses infecting to the bacteria also, and there are viruses infecting to the humans also.
Okay.
So my work was on papain like cysteine protease, and robustly I started doing the cloning, but not in my first year. So in my first year, I started doing literature reviews and helping my. In the beginning, I started helping my seniors. So one day, my boss called me and she said, like, she gave me the patient sample and she said, like, isolate the RNA from this and do the sequencing of it.
Is it during the PhD work or master's work?
No, it is during my PhD work, but I wasn't assigned to my PhD topic.
So you already in a contact with your guide.
No. After joining in NIV for the PhD, so within first year there wasn't topic wasn't assigned to me.
PhD first year.
Yes. So at that time I started isolated RNA from the patient sample. Then I did the sequencing. So I did that within the first attempt. So she was happy. Then she gave me the clone to make a clone of RDRP, that is RNA dependent RNA polymerase. It is also non-structural protein and it is required for the viruses to in replication.
What is the difference between structural proteins and non-structural proteins?
Yes, structural proteins are like wrapping things. So after formation of the genomes, it will be wrapped into the capsid proteins, and then that capsid protein will egress from the cell.
And non-structural protein, they don't wrap in capsin, right?
No. There is no need to go with the protein. For the viruses, what they choose non-structural protein should be expressed inside the cell only, and not outside the cell. So while egression, there will be the only genome and the capsid protein.
Can you give us a sequence of genes present in ORF1? Okay.
So ORF1 is consist of methyl transferase. Then there is Z domain, then PCP domain, then there is macro domain, then helicase and the RDRP domain. So methyl transferase is necessary for the capping.
Is there any specific reason you chose the PCP? Because already you have worked on the RDRP. So why was the decision to work on the PCB other than, because already you're working on the RDRP. RDRP is also one of the important enzyme in case of hepatitis virus replication. Why you chose to be to study on the PCB?
Okay, so RDRP, I did the cloning of RDRP, but I was kind of naive for the virology.
So once again, every enzyme present in the ORF frame is similar as all other viruses and all other systems where they work.
Not exactly. So again, with the host strategy,
Considering into the mind, the viral evolution, you're saying that it is continuously evolving. The rate of evolution is very higher as compared to all other species. And it is transitioning from the plants to animals, animals to humans. Okay. So considering that aspect, going from one category to another category requires a lot of I don't know what to say, but maybe a consciousness kind of thing comes here where it is infecting and trying to get the information from the host, and in the next cycle it is deciding how to infect that particular host which has been already infected. So it is having some kind of data in the evolution process.
yes.
So considering that into mind, can you explain that the enzyme chain which is present here, which is responsible for the replication, is as similar like PCB, which is papen-like system protein present in the papen, and the PCB present in the virus are similar, or there is some sort of difference?
Okay. So structural means with the structure it will be kind of similar, but the sequence will not be the similar. It is like child and adult. Okay. So child is mini adult. So it will have all the.
Do you believe that single mutation will alter the function of a particular enzyme?
May and may not be. So it depends upon where it is actually that mutation has happened. It is if it is in
See, if you consider animal system, if you consider plant system, they are defensive. Okay, they can have their machinery to defend the viral infection. Same with the all other microorganisms. So every time it infects, in case of animal cells, they are having memory. Okay, so again having memory, still you are getting infection. It means that something is changing day by day or routinely in the virus that has changing some sort of functioning in the enzyme that is not accessible for animal or plant cells to deal with it.
Okay. So in case of RNA viruses, this RDRP, it doesn't have the proofreading activity like the host cells or the higher organisms have. So they robustly make the mistakes. So every mistake can be a new mutation, new virus, but we don't know which one is going to come outside the cell and infect the other cells. So that RDRP making mistakes is creating more mutations into the viruses. So it is hard to cure the diseases which are actually due to the kind of RNA viruses. So in case of even HIV, we don't have the actual therapy or the vaccine or the medicine for that. The reason is the same. So it changes robustly. It makes the mistake, it changes, it mutates continuously.
I'm quite curious about knowing that how 1,000 genes fight with the billions of genes.
Actually, in case of RNA, it is not thousands, it is just 10, 12 genes actually. So 10, 12 genes, they will combat with 30,000 genes. So the best part is they change their special orientation. So same enzyme can work differently. So after changing the special orientation, it will show the different strategies or different
The synthesis of proteins in case of animals is as similar as the virus.
synthesis of protein.
Like we have polypeptide, then there is a globular structure, then there is a helix structure, different type of structures we are having. But again, considering the evolution, we are having a few structures that assemble during the protein synthesis. It is as similar as virus, or virus have a different type of synthesis of proteins?
No, they will use the host machinery only for the protein synthesis.
But the proteins which have been generated from this gene, like helicase, RDRP, are from the virus, no?
Yes. But they will be the very simple. They will have the active sites, they will have the specific domains, and they will work. So just changing the spatial orientation, they can have the different functions. So that was the reason in case of plant viruses, the single polyprotein will have the different domains. Like suppose we consider methyltransferase PCB. So in case of suppose we are taking the examples of plant viruses, the polyprotein just will change its orientation and it will change its functions. So like that. So PCB mainly, but during the might be again with the evolutions.
So my question was why you selected PCP.
Okay, so I was naive for the viruses and virus field. So actually, my boss gave me this topic.
Oh, it was sudden.
It was sudden, but one more anecdote was there. When I completed the RDRP clone, there was one MSc student. She was working on this PCP. So PCP clone was there already in my lab, which was around 1.8 kb. So she was comparing the human PCP human viruses, hepatitis E virus, which infects the humans, as well as hepatitis virus, which infects the swine. So she was trying to compare both the proteases, their efficiency. So my boss again told me to help her, and she was just for a.
So this was happening in the PhD second year.
Yes, still my topic wasn't assigned. So every time I was always second choice. So I came, I completed the work which was given for me.
Why is it so?
No, I don't know. So, but that was the pattern. So again, further I will tell you the more things about this. And after that, she left. So that comparison topic was given to me. So I gave I completed that assay. So specifically that assay was like de-ubiquitinations, means post-translational modifications and its involvement of PCP, a protease enzyme, in post-translational modifications of viruses. But what she was doing outside. She was doing it with the clone and not inside the cell. What should be in it will come later on. So she was doing that mm, in vitro studies kind of thing. So I did. So I took that project and I did.
So what was your conclusion? What was the difference between swine and human PCP?
So human virus PCP was more robust in deconjugations method as compared to
Deconjugation of what?
deconjugation of see, there are there are more than four kind of post-translational modifications like sumylation, nidylation, isgylation, then phosphorylation, methylations. So these are the post-translational modifications for proteins to assign their work inside the cells. So what PCP or the protease is doing, so whenever there is hindrance in their replications, they will try to deconjugate that part. They will cleave that assigned their locations or suppose sumylation is done with a specific protein. So the PCP will remove that sumo tag. Now the protein will be confused.
When does it happen? During defense mechanism?
Inside the cell, it is happening.
So it is removing the location tags from the proteins while there is activation of defense system from the animal cell. And some kind of diversion is there in the animal cell, and so there will be no death of cell or kind of defensive mechanism will not be there.
Yes.
Okay.
So
PCP is the main protein actually in this case.
Yes, it was a little bit known for this. It does the deconjugations of the protein tags. And she was trying to compare the human virus, hepatitis E virus, as well as swine hepatitis E virus.
So in case of humans, the enzyme which is responsible, that is PCP, is responsible for deconjugation, and the rate of deconjugation is very higher in case of humans as compared to the swine.
Yes.
And even stability, in case of stability, because defense mechanism is activated, so there is need of stability of enzymes which have been produced by the virus. So stability is more in case of human PCP. Okay.
So once my project was assigned or my PhD topic was assigned, so what we did, we again started cloning the PCP, but now we took the extended version. So the clone available in my lab was around 1.8 kb. So we extended it to 2.4 kb as well as 2.9 kb. So from the PCP, there is one proline-rich region and as well as microdomain regions. So we are trying to find out their role also while polyprotein processing as well as inside the cell, what they are doing with the help of PCP. So proline-rich region is a kind of, it is providing the flexibility. So suppose if enzyme is there, it needs to move here and there. So that proline-rich region, it will act as a hinge region. It will provide the flexibility. And macrodomain, during the evolution, again, hepatitis E virus, that it might have taken from the histone protein. So there is a human histone protein H2A. So it shows the similar kind of similarity with the macrodomain. So what generally viruses do
Macrodomain is very well studied.
not well studied, but its role is a bit known. So again, macrodomain will help the virus in fighting with a defense.
But how
That in
histone proteins are part of nucleosome, no?
yes.
Then how that macrodomain synthesize histone proteins and how they help?
No, it is kind of mimicking the histone-like protein. There is the domain of macrodomain which will show the similarity with the.
But why it produces histone proteins? Why it is needed?
A bit of it. It has a role in innate immune system, like interferon regulatory factor IRF3. So it removes the phosphorylation of IRF3, so that the innate response, which is of the interferons and interleukins, it will not be expressed. So they are using that domain for their kind of replication settlement. So
Okay. I have two basic questions. First is, there are different types of hepatitis virus. Starts from A, B until E. So what is the basic difference between them? And another question, how does a virus decide which cell to infect?
okay.
There are two questions, so I just would like to know about you.
Okay. So hepatitis A virus is RNA virus. Hepatitis B virus is DNA virus. Hepatitis C virus is again RNA virus. Hepatitis D is DNA. Hepatitis E is again RNA viruses. So, but hepatitis A have the different family, E have the different family, and hepatitis C have the different family. So hepatitis C virus comes from the family of dengue virus, West Nile virus, yellow fever virus, Japanese encephalitis virus. Hepatitis A comes from the Picorna viridae family. So foot and mouth disease of the animals you might have heard. So it comes from that type. And again, hepatitis E is more interesting. So it has the separate family for the single virus. So first of all, in the beginning, scientists try to put them in different families. So they thought it will be like hepatitis A virus. So they try to put in Picorna viridae family. Then they realized, when the genomic studies was done, so it was different from the hepatitis A virus and also from the hepatitis C virus. So separate hepiviridae family was created for this virus. And more interesting for the hepatitis E virus is it can infect humans, it can infect pigs, it can infect mongoose, it can infect ferrets. So it has the host range, wild host range in case of animal. And this is the only virus from the hepatitis family which will have the animal reservoir.
You mean A1, right? Hepatitis A.
Hepatitis E virus.
Okay.
So all other viruses, they don't have the animal reservoirs.
So, I mean, what was the reason of you choosing specifically hepatitis E?
So I don't choose hepatitis E virus for my PhD.
Then guide provided.
Okay, okay.
Given by my guide.
Okay.
But I found more interesting than other hepatitis viruses.
Can you tell us how does hepatitis caused by the virus, exact mechanism? How does hepatitis is developed in human body?
See, the RNA viruses will have the different strategies, and DNA viruses will have the different strategies. So, in general, there are multiple ways to infect the cells. Some will be the receptor dependent, some will be the receptor independent, some will be directly phagocytosed or pinocytosed. In case of Ebola viruses, they are directly pinocytes.
Can you explain phagocytosis and pinocytosis?
Pinocytosis is like it will come with the liquids. It is like drinking. So pinocytosis means cell is drinking something.
Okay.
And phagocytosis, it is like eating something.
Okay.
So again, viruses have the different strategies. Within viruses also, they try to get help from the host proteins. So in case of SARS-2 or COVID, what we have faced as a pandemic, so it has the furin as a protease present on the membrane of the cells. So that furin will cleave the some of its part of the capsid, or you can say glycoprotein part. Then the hydrophobic domains will open, and then it will attach to the membrane, and then virus will come inside the host. So these are the different strategies, but they need the hydrophobic domains, because as a plasma membrane is made up of lipids. So hydrophobic domains will help them to come very easily inside the cells as compared to the other.
Okay, now let's move on to the gene. Which segment you cut down and utilize for the study?
Okay, so within protease PCB, we try to increase its domain, so that in 3D orientation or 3D configuration, we will find more interesting results. So we cloned up to 2.4 kb and 2.9 kb genome.
These segments, which genes were there?
Protein rich region was there, and
Which sequence?
in 2.4 kb clone, protein rich region was there, and in 2.9 kb macrodomain was there.
Only macrodomain?
Yes, up to the macrodomain, means from methyl transferase to the PCB to the macrodomain, including protein.
Or starts from where?
Again, from the methyl transferase, it will end to the protein rich region. So inside there will be the methyl transferase, then some Z domain, then again PCB and protein rich region.
Okay, PCB and proline rich is 2.4. And up to macrodomain, it is 2.9. Okay, so 2.4 consists of methyl transferase, then X domain, PCB and proline rich. And 2.9, it is having methyl transferase, X, PCB, macro, and NCF.
So it is Z domain, not the X domain.
Z domain. Okay.
So it is the extended version of methyl transferase.
And this Z domain is responsible for histone protein production.
No, that macrodomain, not histone production. It kind of mimics the domain of histone H2A protein.
H2A, Z domain.
No, macrodomain.
And what Z does?
Z is a kind of extended version for the methyl transferase. Again, it will help.
Extended version is conserved region?
It is conserved, but not specifically. It
Brief about which domain from this 7,000, about 7.2 kb region, which regions are more involved during the evolution process?
Okay, so what we have said, like that in between the Z domains or protein rich domains and the macro domains, they are more participate during the evolution.
Protein rich and
The macro domain.
macro, and Z?
Z is also
Okay, Z, macro and protein rich are the part of evolution process.
kind of.
And methyl transferase, PCB, helicase and RDRP, they are similar as all other enzymes.
They are highly conserved, so they are more necessary for the replication.
But slight mutation is there.
The slight mutation is there.
Are these genes which are Z, macro and protein rich responsible for the changes in the methyl transferase and helicase, RDRP and PCB?
No, actually they are. See, main domains are methyl transferase, PCP, and kind of macro domains. But they are trying to absorb the mutations in between. So they are like acting as a shock absorbers. So while creation of mutations, more stress will not come to the methyl transferase, PCP, macro domains.
Okay. Okay, now you are having two domains. Two domains of genes. One is 2.4 and 2.9. So give us a brief about what was the conclusion or kind of research you did on 2.4 kb and 2.9 kb genes.
Okay, so we cloned it in bacterial expression system.
Bacterial transformation.
So we use the plasmid, which will be useful for the bacterial expression.
A vector, right?
It is vector. It is kind of vector. So what we first we select the expression system which we want to express, like bacterial or insect. Or
Select that system, expression system.
it is available in the market, so you can purchase a vector which will be of the bacterial system.
How you decide that I want to express in bacteria or any yeast or something like that? How to decide it?
So bacterial expression will be give you more kind of yield as compared to the mammalian and insect cell lines.
Give us a brief about how you cut this section and incorporate it into the plasmid, and how bacterial cell. Maybe bacterial cell will also be, again, it is having virulent property. Okay, it is not a normal gene. Again, these enzymes which has been produced are not the normal enzymes. So how that competent cell, bacterial competent cell, do they have defense mechanism, or it was very easy to do transformation?
Okay, so cloning of PCB was very kind of challenging thing, because we are trying to express viral protease inside the bacterial system. So already it was becoming toxic. In general, protease is toxic to the cells. So we are trying to clone the viral protease inside the bacterial system. So it was again kind of more toxic for the cells. So first 2 years I was struggling with the cloning part only, because bacteria wasn't allowed to
Competent bacteria.
a competent bacteria. Though it is competent for the cloning system, still I wasn't getting the proper protease domain or proper protein of protease, or the enzyme of the protease. And so we use the different strategies. We lower down the temperature of the bacteria while expressing the induction, while doing the induction also. We use the different cell lines like P-lysis, limo cell lines, BL21 cell lines. These are different cells which can be used for the protein expressions. So
Temperature helps.
yes, it helps. It lowers down the metabolic pathways of the system or the bacteria. So now the challenging clones can be. It is easy to transform the that specific clone inside the bacteria and make it just create its soothing effect for the bacteria, so that it will take inside the clone and it will express as a protein.
But it will similarly impact on the enzymes which has been produced from your targeted gene. Again, you are lowering the temperature. It means that the defensive section, or maybe the proteins which are required for bacteria to produce, the production has lowered down. In the similar way, the production of viral proteins also will be reduced down.
It will reduce down, but we are increasing the time span. So for the overnight, if you keep
So lowering temperature, increasing time span.
Yes, to lower down the temperature and increase the time of induction, so that you will get the enough amount of protease enzyme, and then we can do the assays.
Can we compare the speed of production of proteins from bacteria and virus? I don't know, but I think bacteria have rate of production higher than the bacterial cell system.
In general, the specific enzymes of the viruses are more robust.
What do you think of a rate of production of proteins?
Not exactly. I don't know about the rate of production.
Because lowering temperature, increasing the time span, it will affect both viral proteins as well as the proteins from the bacteria.
Yes, and that is the.
If you are saying that there is successful recombination or kind of more plasmid production or proteins production, it means that viral proteins have higher rate than the bacterial cell system.
Yes, obviously, because we are what we are doing. We are intentionally putting the proper vector system inside the bacteria, which will express proteins only of our interest and not other.
Or maybe the virus proteins do not have impact of temperature.
No, they can have the impact of temperature, but what we are doing, we are lowering the temperature for what? So that the bacteria.
You're talking about defense system.
Not defense system, but bacteria will not what kind of oppose the
Responsiveness.
responsiveness. It will not oppose the virus system or virus protease, and it will kind of
Okay, see, what I am imagining is that because of lower temperature, higher time span, consider time as a different entity here. Focus only on the temperature lowering. Lowering on temperature, it means that time has been slowed for the viral proteins. Now viral proteins have entered. Now the time slowness for the viral proteins is different. The time slowness for the bacterial system is different. So that the reason the defensive or maybe responsive proteins which are necessary to produce against that viral protein are not produced by the bacteria. The rate production is very low as compared to the viral proteins because they maybe have less affection towards the temperature as compared to the bacterial system.
Okay. So it is not specifically like that, or maybe it may be like that, but we are providing the lack of, inside the expression system. We have the lack operon, so we are intentionally inducing to produce specific enzyme only and not other things.
Really like operon?
So lack operon is actually a set of genes where the kind of it will produce under the stressful conditions. When the glucose is not available, then the bacteria will shift from the pathway. It will shift the pathway. So what scientists have done, they have deliberately used the lack operon system for within the vector to express your gene of interest or your protein of interest.
Okay, now lack operand is a part of indication process. Okay, so it indicates. Okay.
It acts as a kind of indicator.
Yeah, whether transformation has occurred or not.
So there are also this thing, blue and white screening, right?
Yes. It is blue and white screening, but it is the again different scenario to get whether your clone is not exactly shifted inside the plasmid or not. So blue-white screening is used as a indicator or as a reporter for the confirmation of cloning.
Okay, how was the result of blue-white screening?
Yeah, it was
Similar as all other studies or different?
because the principle remains same.
What was the percentage of blue and white cells?
Means it depends upon whether your clone is ready or not. So if your clone is perfect, means it is ligated properly, then there will be with a more amount of your expressed.
That's what I'm asking. What was your result?
We can get good amount of clone. So it wasn't like, but there are, okay, so you said it for the blue-white screening, but there are the chances some blue colonies may change later on into the white.
How is it like?
I don't know the exact mechanism why it happens, but it happens. So sometimes they can.
There are chances of getting false.
Kind of false positive thing.
Maybe bacterial system has activated.
Or maybe they have adapted.
No, they have degraded the gene of your interest.
Might be.
Well, again, they are also part of evolution process and part of survival system. Okay, so now let's move on to the 2.4 and 2.9 kb gene region. Now you are transferred 2.4 into the plasmid and now infected inside the cell.
The 2.4 and 2.9.
And you did screening. So further you did SDS and all.
Yes, I did that.
What were the results?
Yeah, I got the exact amount of protein which was necessary. Okay.
See, right here in 2.4, there are different genes like methyl transferase, Z-domain, PCB, proline rich region is there. So you targeted right here PCP, correct?
Yes.
And you check the functionality of PCP only, or methyl transferate and Z-domain as well.
I am trying to find out the function of the PCP, but along with the extended version.
So it was all together. You wanted to study that interactive mechanism of PCB methyl transferase together with proline rich region.
Yes.
Okay, so that kind of there. And in case of 2.9 kb microdomain region?
Yes, microdomain region is there. So what? So again, I have clone from hepatitis E virus of the humans as well as hepatitis E virus from the swine. So actually, I have 4 clones.
Is there any difference between studying the clone genome and the original genome?
Yes. There will be the difference again. Because I am specifically targeting few proteins or few enzymes of the viruses and not the total full genome. So full genome study will be done inside the mammalian cell line.
Your clone consisted of all the original proteins?
Or not all the original proteins.
That is
Because,
not exactly the same.
helicase is not
a
there.
small
RDRP is
portion
not
of
there.
full genome.
Helicase,
No, I'm talking about your clone.
yes.
That clone is actually part like
No, I'm talking about that original clone that you are I think that junior was studying on.
Yes, but it was again with the methyl transparate and PCP. So it was smaller than the actual.
So it was whole onto the 7.2 kb base pair.
So actual total genome is 7.2 kb.
Original.
In
case of clone?
In case of clone, we are trying to clone the different domains or different enzymes of the or full genome. So we are not taking the total 7.2 kb genome. So with the 7.2 kb genome, we will go in mammalian cell line. So now we are expressing it in the bacterial system only.
So you're focused on the 5.3 kb.
5.1 kb of the ORF. So
genome which is responsible for synthesis of that particular enzyme.
No, there are lots of enzymes. Helicase is there, RDRP is there, macrodomain is there, PCP is there, methyl transfer is there. So he's doing the interactive study. Like together, what happens together with methyl transferase and PCB and protein rich extended region. And he's trying to know how macrodomain, helicase and RDRP work together. And also he had eliminated all other genes also which are not part here, that is capsid region proteins. So right now,
More of a structural
2.4 plus 2.9, 5.3.
So helicase and RDIP is not part of my current study. Okay, so
you target only PCB.
Yeah,
I have targeted PCP with extended version, but I haven't taken the helicase and RDRP region.
Okay, so you did transformation of both 2.4 and 2.9. Blue white screening was done, and what was your conclusion? What is the conclusion for 2.4 and 2.9?
That is what I am saying. The macrodomain part is not involved in 2.4 kb. So 2.9 kb macrodomain part is there.
No, I'm talking about the blue white screening results. The SDS and all other things that you carried out after a kind of extraction process. So how was that? What was the difference between the 2.4 and 2.9 functionality of proteins? How they interacted with each other? That kind of.
Okay, so you are asking me about the assays.
I'm talking about the production of enzymes. So like if you consider 2.4, which enzyme produced in more amount? How was the functionality of enzyme? In case of 2.9, which enzyme was more active, which you were extracted, and how was the functionality of enzyme? Was it similar as clone or it was different?
No, it is hard to find out which protein is expressed more because it is total single kind of polyprotein for this thing. So it is hard to find out which protein is expressed or more or less. So we have that fragments of different proteins. So interesting part was like we have find. I didn't get the actual polyprotein like up to the PCP or up to the protein rich region or up to the macrodomain, but I was getting the different fragments of it. So I never get the intact.
No, earlier you said that it is a in virus, polyproteins work together. It's like they're changing their function. Yes. It's like kind of maybe hide and seek. So considering that, it is okay that you will get some of the polypeptides, you will not get some of the polypeptides. But what my question is that what is the exact conclusion of SDS of 2.4 and 2.9? Which proteins were there?
What he means is like whatever the assays or the tests you have conducted during your research. So what the result of that assays or test,
Yes.
and how they like contributed the final conclusion of your research.
Yes. After
If I'm right.
Correct.
Okay, so after expression we did the assays for it. So till the SDS page we were getting the fragments only. So we don't have the specific different. We don't know which part is cleaved by the virus, and also the protease, it has the self-catalytic activity. So there are chances might be the same PCP is cleaving the other domains, or there might be the chances of bacterial protease cleaving the my polyprotein part.
Okay.
But there were PCP in case of 2.4 and 2.9 macrodomain.
Yes. There was,
So you assured that there are these proteins.
yes, I was, because the total length was, which I was expecting, I got the total length, including my tag.
Okay, so there was a primary confirmation that these proteins work.
Yes, this protein expressed. Now the assay will come. So similar what I have done with the 1.2 kb gene, I put the deconjugation study.
1.2 kb gene means
1.8 kb.
okay. Again, you produce 1.8 kb gene.
No, it was already, but this assays which I did with the 1.8 kb gene.
Okay, what was that segment?
Again, the deconjugation assays.
Okay, which enzyme we involved doing this?
So there are artificially
1.8.
1.8.
no, I did with 1.8 kb before.
Uh,
Now I am doing it with the 2.4 kb and 2.9 kb.
okay, so what was the result? What were the results of 1.8?
Okay, so finally we did the assays, deconjugation assays, which we had done with the 1.8 kb clone.
Okay.
The 2.4 and 2.9 we did.
Can you give us a brief idea about the 1.8 kilobase per region?
That was with methyl transferase up to PCP.
Methyl transferase up to
PCP and not included.
Not proline rich region.
And not macrodomain.
Okay.
So we found out that extended version is giving more efficiency for that specific enzyme to deconjugate. So
means 2.4 region.
2.4 and 2.9. They are good in deconjugating assays.
Why is it so?
Again, what we have hypothesized that extended version might be giving more conformational changes for the polyprotein processing.
Okay.
And after that, now one more thing we did. Also, we want to check the PCP with the host protein interactions, means virus host protein interaction.
Okay, before that, tell me the differences that you observed in the 2.4 and 2. Regarding the conformational changes. Now, what you said in case of 1.8, the conformation changes were very less because extended region wasn't there. But in case of 2.4p, extended region is there, that is proline rich region. In case of 2.9, there is a macrodomain additional to proline rich region. So, what is the difference that you observed in the proline rich region and along with proline rich macro region?
Macrodomain region was giving more enzymatic assay results, good enzymatic assay results. So again, what we had hypothesized, that is confirmation is giving more efficiency for the PCB domain to work out for the deconjugation assay. And same thing we did with after these clones, we try to achieve same results for the animal cell lines. So inside the cell lines. So now we are going to.
No, what about the deconjugation process? What type of changes were seen in case of 2.4, 2.9 and 1.8 regarding the deconjugation process?
Yes. These are for the deconjugation, we used substrates, artificial substrates, which were already somalylated, nidilated, isgylated, and ubiquitinated. So
what does it mean by ubiquitinated somalylated means? Already modifications were there. Means that proteins were tagged already.
Tagged with artificial substrate.
Okay.
So first what we did with the clones and artificial substrate, we showed it in vitro. Now we are going to show it inside the cell with the full genome now. Okay,
so in case of after transformation, you just made a conclusion that extended regions are responsive for conformational changes and
affinity chromatography, we purified this particular enzymes and then we did assays. Deconjugating assays.
Which enzymes?
That is the PCP. So for the whole genome, we need the specific cell line which will grow robustly. So generally, hepatitis E virus, it has very less replication efficiency inside the cell line. So hardly 10 to 20%. So out of 100 cells, only 10 to 20% will have.
Now just tell me which sequence you gonna use in the stable animal cell.
Now we are going to take total 7.2 kb genome. Whole genome. But one more interesting thing is that directly clone is not infectious for the cell line. So what we were doing, we were linearizing the genome Then we were doing in vitro transcriptions. We were preparing RNA, and then that RNA with lipofectamine reagent, we were in fact transfected to the cell line. Okay, so directly virus infection to the cell line is not possible. Direct infectious clone is also not possible to infect the cell line. So we have to use that strategy. It is like mRNA vaccine. So you are from the DNA, again you are making the mRNA, you are packaging with chemical like lipofectamine, and then you are infecting that packaged bundle to the cell line.
Okay, so you're gonna create the negative strand of RNA.
As an intermediate.
That negative strand will be converted into positive strand inside the cell.
And now replication will start. So for that, again, as I said, I was always second choice because those were two projects where already, again, MSc students were working and the project were too big to handle. So I was helping them, and later on, totally they were given to me. So one aspect was to develop a stable cell line for hepatitis E virus replication. So how we have done that?
It is because cell lines are not available for that particular virus.
The virus is not that much infectious to the cell line. Cell lines are available.
Okay.
But virus is not that much.
So you are saying that you made effort to make stable cell lines.
Yes. So what we did, we removed the capsid region from the 7.2 kb, and we put neomycin resistant genes inside the genome actually.
Okay. Instead of capsid proteins.
So now what producing? Capsid protein. So now what will happen? Now the genome will replicate inside the cell, but it will not come out as we have removed the capsid. So now it is not going to pack into the packed as a viron or virus particle, and it is not going to come out of the cell. So with the cell division, all the replicated genome will be again divided into the cells only. So with this strategy, now what we did, we prepared the stable cell line. One more thing we did, we increased the poly-A tailing of the actual genome.
Give us a brief about how to make a stable cell line.
Okay.
And why it is so difficult to make stable cell line?
Yes, it is difficult to make a stable cell line, and the reason is already the virus is not that much efficient to replicate inside the cell or infectious to the cells. So the first target was instead of virus, you are using the full genome, infectious, not infectious, but the complete cDNA. So from that cDNA, you are preparing by in vitro transcription, you are preparing the RNA, and that RNA you are going to transfect to the cell lines. But instead of again full genome, we have removed the capsid region. So we put neomycin resistant genes. So again, it was the proper kind of R&D. So to remove almost 800 nucleotides, we removed from the actual genome, and we put neomycin resistant genes. So we have created the restriction sites there.
So my question is that why there is need to put efforts to make stable cell line? Is it because of the virulent property of that genome, or is there any more to it?
Generally in the field of virology, we tried to check the cytopathic effects.
Whole genome is toxic to cells?
Whole genome is always toxic to cell, but the thing is like we are trying to develop, generally what virologists do, they try to develop a cell line where virus will grow robustly, and now it is very easy to study that particular virus.
Making stable cell line means do not allow cell to die.
Not already we are using hepatoma cell line, so already they were cancerous cell lines.
Then why there is need of efforts to make stable cell line?
Yes, to understand the virus strategies, to study the viruses, you need either animal host, proper animal model, or tissue culture model. Means we need both the things. We need the tissue culture system where virus will grow robustly, so that we can study the interaction of the viral protein with the host proteins. So that was the one more strategy. And again, we did the poly A tailing for the particular genome. It was already reported. If you increase the poly A tailing, the replication efficiency of the viruses increases.
Now the genome that you have used, it is from the methyl transferase to RDRP.
Now it is total 7.2 kb.
Now you replace the capsid producing genes with neomycin resistant genes. It means that from methyl transferase to RDRP with neomycin resistant gene.
Correct, exactly. So now we have the full genome, but the changes we made, it is in the cDNA. And from that cDNA, we prepared the mRNA, means actual virus RNA. Then we packed it into the lipofectamine reagent, and then that we transfected to the cell line to study and to confirm whether it is a cell line is stable or not. So inside the medium tissue culture for the cells, there is a need of medium to survive, to eat something. So inside that medium, we put neomycin. So those cells which were not with the neomycin gene, that will die, and the cells with the neomycin resistant gene will survive. So ultimately, your full genome or your actual viruses, cells with the viruses will survive.
Like a hydroma technology.
Hybridoma technology. Not exactly, but we can say we modified the virus genome and we put so that continuously it will replicate inside the cell. So that, and one more strategy, poly A tailing. So again, we make the changes in the actual cDNA. Then we prepare again in vitro transcription with mRNA. Then again we infected that poly A tailing. So how we concluded that it is the stable, we have made the stable cell line. So for that, again, particular domains from the ORF1 region, we cloned separately. We cloned methyltransferase separately. We cloned helicase. Helicase were already there. Again, I prepared the RDRP genome. I prepared the macrodomain clone. So from these clones, again, I expressed the recombinant protein. Those proteins were mice immunized. And from that, polyclonal sera was used for the Western blotting. So that whenever I will do the SDS page and Western blotting, if I found the particular antibody expression is there, means what we can say, particular that stain is there on the X-ray film, we will develop the blobs of how we in old days we used to develop the X-ray film after photo imaging. So similarly, in the dark room, we developed the X-ray films. We used antibodies of the viruses, means reproduced from the viral clones or viral proteins. And for the confirmation, we used that. So one more thing we did again real-time PCR for the particular domains expression inside that cell line. So we proved with real-time PCR, means at the RNA level, we can say, and at the protein level with the Western blotting. So we did both the things for the stable cell line also and for the poly A tailing also. So out of that poly A tailing, I used to check the virus and host protein interactions. So in this case, virus protein was the PCP. So how PCP is interacting inside the cell line. So we found out that. So again, we did.
You did poly A tail to every gene?
A total genome.
So when you are studying the interaction of PCP, that PCP gene was alone or with poly A tail or with whole genome?
Now it is with the whole genome, means poly A tailing.
But you mentioned about you clone different genes.
Yes.
Different genes. And what you did with it?
I raised antibodies against them for the Western blotting.
Okay.
And the first thing I cloned the macro up to the macro domain. That was for in vitro assay.
Production of antibodies against a particular gene, it means that gene is pathogenic.
That gene is present inside the cell. Means I I cloned the viral domains. Okay.
If it is a single gene, okay, for example, PCP gene is there. If it is transferred inside the any animal cell, does it activates the immune response?
Yes, it will activate and protein will be present there, right? So I am expressing the full genome. It means that all the proteins, non-structural proteins, as well as now in case of neomycin, there is not capsid region. So I am not generating antibodies against antibodies which are useful for the capsid protein detection. I am not using that. I am using antibodies for the ORF1 region, which is non-structural proteins region. So what I had done, I used methyltransferase, a macrodomain, then proline-rich regions. Separately, I cloned them. I expressed the recombinant protein. That recombinant protein I injected into the mice for the polyclonal serum.
My basic question is that if you're dealing with production of antibodies using a particular gene, now my question is that just assume there is a cell and we have injected the particular gene, that is PCB gene, we have injected. What will be the response of that particular cell? Will it degrade the PCB gene or it will create an immune response?
It will create an immune response because now we are not injecting inside the cell.
I would like to know the reason behind it. Why it will make efforts to create antibody instead of degrading it.
See, in case of animals, we are injecting, so it is not going inside the cell, right? So it is circulating into the body. So adaptive response is going to come forward.
Consider there is a tissue of a particular muscle tissue or any tissue you consider in your case. Now we have injected, consider we have injected the solution of your gene of interest, that is PCP. What will happen?
Now the response will be the innate immune.
My question is that why immune system is involving in it?
Which one?
Because does that particular cell or group of cell, do they consider that as a foreign body and create immune response, or will try to degrade sequence because the gene sequence is very few base pair sequence? So instead of going to invest more time into the production of antibodies, it can simply degrade it.
So degradation is a part of the immune
And
response.
another question I would like to add into it, if it is creating antibodies as you are mentioning, it means that individual gene that virus has produced has some immunological response because it is having some virulent properties.
Yes, always there will be the virulent properties. But the thing is, inside the animal, if we inject, the adaptive immune response will be there. So B cell, T cell response will be there. But when we consider the tissue culture or the animal cell line, not the animal, now there will be the innate immune response. So degradation is the part of innate immune response. So inside the cell line, antibodies will not be generated. For the antibodies, you need the B cell, T cells, and other. But you can consider means if we brought more broad spectrum, we need dendritic cells, we need macrophages for the antibody response for the generating of antigens or antibodies.
See, antibodies are interacting, it means that cells are knowing about it. Okay. Yes. That is the reason antibodies have been produced. You are saying in Western blot, antibodies interacted with that particular gene. It means that antibodies are having specific region that is designated towards that particular antigen. Yes. It means that gene PCB or maybe all other genes are virulent to the animal cells.
Yes. See, there is a confusion. What we have done in the animal cell line, everything was expressed, right?
Okay.
It was full genome. So after that, we extracted the cell line cells. We lyse the cells. Now what is happening? Once we lyse the cells, everything will come out. Okay. So we run on the SDS pages. So Western blot has the what we can say SDS page is part of doing Western blotting. So first we did the SDS page. Later on, from the gel, we brought it onto the nitrocellulose membrane or PVDF membrane, and then we treated with the antibodies which were already generated from the mice polyclonal serum.
The basic question is that why there is need of antibodies to interact with them. The main thing is that antibodies are generally present at the cell's extracellular structure, correct? Or maybe produced from one side and it will attach to a certain region and will interact with the viruses, or maybe flowing inside the blood. In this case, I don't know about it. You mentioned
Antibody generation is the work of B cells.
but the response has been generated by cells.
See again,
Who has given message antibodies to create it?
see,
The infected region.
what I wanted to tell you is that the infection is for the cell line of the full genome.
Yes.
And with the clones, we injected into the mice to generate the polyclonal sera. So that sera, and after the SDS page, we are going to expose that polyclonal sera to the cell acid. So inside
my
the
basic question is that why our body putting efforts to make antibodies against to that particular gene? See, it hasn't produced the protein. It is just a sequence. Still, cells are taking efforts. Again, it has started from somewhere. Okay, it has infected somewhere or it has ingested somewhere. From that, response has been given to those cells who are going to produce the antibodies, and from that they are traveling and interacting with virus or the particular gene, and that gene is going to degrade or something else is going to happen. Correct?
No, I think you are confusing between the actual. We haven't infected the virus to the whole animal model, or
No, I'm assuming.
No, but actually it has to be like that. Attenuated viruses or some like, because means what his point is like, until unless the our immune system will understand antigen is actually viral proteins.
yes,
Okay, not the genome.
yes,
Right.
That is what.
So if at all you inject genome, I guess that's the point. Yeah, that's his point.
But
if
I
at all you
didn't-
inject genetic material, understood? That is what his concern.
Okay,
What he wants
but
to understand.
I'm not injecting the full genome to the animal models or the mice.
Now I'm talking about that single gene PCB.
Yes, but obviously, because from suppose not exactly PCB, but anything that non-structural protein was there, I expressed as a protein with the recombinant protein expression. I used the cell lines.
Actually, he got antigen synthesized, and that antigen has been introduced to
that
the
protein
animal
is injected
model,
into
and now
the-
that antigen expresses the immunological consequences afterwards.
Yes.
And now, exactly, triggers the immune response, and so he got the antibodies into the serum of the animal model.
So to generate the just polyclonal sera, we used the mice, and our aim wasn't to infect the total genome with the mice.
Okay, so your next study was inside the cell line, robust cell line.
But first, we have to confirm whether we have prepared the stable cell line or not. So for that concern, we need the antibodies produced from the mice sera, because inside the cell there will be the again proteins expressed.
So in case of mice sera, you injected as an antigen. Okay, so extracted proteins. Extracted proteins were injected inside the mice, and then the immune response was generated, which is correct. And that sera you used, that antibodies you use in the interaction with the kind of interaction study.
For the animal cell line.
Now we are here trying to understand the replication process. Now you're going to inject the either whole genome or specific gene inside the cell, and you're going to study that how it is going to interact with all other proteins inside the cell.
Yes. For the animal cell line, we are going to use, or we had used, full genome, not the particular clones. Okay. So again you will confuse with the full genome, but full genome as a mRNA or as a positive sense RNA virus. What is its actual genome is. So that what positive sense RNA, it will go inside the cell. Now it will produce everything, non-structural proteins, capsid proteins in case of poly A deli, but not with the neomycin resistant genes. But the non-structural proteins will be expressed in both the cases.
You use whole genome and also genome with neomycin resistant gene. So two comparative studies are here right now.
So now we established both the strategies, but we used a polyethylene genome for our virus host interaction studies. So what we have find out, almost 600 proteins are modulated by PCP region inside from the host cells to establish itself inside the cells.
How many proteins?
600
600.
proteins.
For
Just from the PCP domain.
from the PCB domain.
Why I am saying this PCP? Because we have showed it in vitro studies that PCP has
2.4.
in 2.4 and 2.9 we have showed deconjugating activity, right? So again we try to put we are trying to check post translational modification which has happened inside the cells which are modulated by the viral genomes now. So in the end we did the mass spec studies and we found out almost 600 proteins.
So you inserted 2.4 kilobase per region, that is from the methyl transferase to PCB, correct?
For the cell line I am using full genome.
Okay.
These clones and their protein.
Give me conclusion regarding the methyl transferase to RDRP with neomycin and methyl transferase to RDRP.
Okay. See, neomycin gene is the actual full genome, right? So that full genome
Capsid region you have removed just.
we have removed, but we have impacted to the cell line, right? So it is remaining.
Because you have removed, so the capsid will not be formed, right? So there you have only the rest of the genome and resisting gene, right? So what his point is, inside that cell line, when the gene is inserted, this virus will achieve its lytic cell when it completes its entire capsid formation takes place. When the gene is get expressed and it goes to the lytic cell triggers what? It will burst the whole cell, right? Instead of that, because he inserted this resistant gene instead of your capsid, so capsid will not be formed. Because capsid has not formed, the cell will not go in. It will be neither lytic cell nor lysogenic.
So
It will be in between. So you have multiple copies in the internal environment of cell, like cell sap, we may say. And you have abundant amount of that RNA. So further he will study on that copies. It is like that. Instead of getting that virus in its complete form and it goes to the lytic cell, so it destroys the host cell coming out of that cell. So what he has to do, he has to maintain the host cell as well as the copies of RNA inside it. So we could study. That's
In case of methyl transferase RDRP.
now there he is studying actual what happens when the lytic cell goes further. And that is the immunological response we are understanding inside the animal model, right? Yeah.
Just I wanted to correct that means lytic and lysogenic cycle is actually not happening with the RNA viruses.
Okay.
So they egress from the cell.
Okay.
So they will not kill the cell.
Okay.
Cell will remain intact, but they will egress from the cells.
Then what is the, like, I do not understand what is the purpose of like not allowing happening the capsid, if at all it's not going, means in the initial phase I understood like it is going to be the lytic cell, that's why you just mutated that particular gene. Yeah, I like I looked like that. So
You give us a proper explanation regarding this.
PCP region is in non-structural protein region, right? Or in ORF1 region. So I am concerning with the PCP's role. So I am not interested in the capsid or the viral egression.
Okay.
So I am just checking the role of ORF, means PCP, which is present in the ORF1 region.
But PCP, it is along with macro, helicase and RDRP.
Yes.
So have you removed that region or it is with that?
No, see, inside the cell, what is happening, we exactly don't know.
No, I would like to know what you did.
No, I haven't removed anything. It is full genome.
Okay, so let me give a brief regarding this. What I know about your experiment. One is with methyl transferase to RDRP, that is combination of methyl transferase Z, PCB, proline, rich region, macrodomain, helicase and RDRP. Correct? This is a whole genome. Plus neomycin resistant gene is there. correct? And second experiment where neomycin gene is not there and whole genome is there.
No. See, how I should explain you.
With experiment.
With the experiment.
Yeah.
See, there is two different aspects, right? So I used clones and their proteins or the recombinant protein expressed enzymes or specifically PCB. So I did with the clones, but clone again expressed as a proteins. Okay. So because I made the clones, I expressed into the protein expression cells. Now I have the proteins, or you can say the enzymes. That enzymes I used with artificial substrates. Now what I showed it outside the cell, in vitro studies I did.
Yes, that is with post-translation modifications.
But those were the artificial substrate, not from the cell line. So those were the just clones.
Yes.
It wasn't their full genome. Now I have used full genome for the cell line. So inside the cell lines, how they are behaving, we don't know. They may be working as a polyprotein.
What was your whole genome?
Again,
Starting for and where it ends.
It will start from methyl transferase and it will end to the 7.2 kb. Means where ORF1 will be there, ORF2 will be there, ORF3 will be there.
Okay. And then?
Then now I'm studying it inside the cell line.
Okay.
So understand how virus will behave inside the cell line. I'm trying to mimic that.
It can be a standard.
Yes, you can say the standard.
Proper standard.
So we developed two strategies with the neomycin resistant genes as well as polyethylene. But for the experiment,
Okay, wait. So neomycin resistant gene, what was the genomic content?
For the neomycin, capsid is removed, but from the methyl transferase up to the ORF3 region is there.
Wait, ORF3, what it corresponds?
Up to it 7.2 kb.
ORF3 responsible for phosphoproteins, correct?
Yeah, yes. It helps in viral egression actually.
And also it is having prolinal rich region.
Obviously, because ORF1 will be there.
Okay, so it is now combination of methyl transferase Z, PCB, prolinal rich region, macro, helicase, RDRP with ORF3 that corresponds to phosphoproteins and prolinal rich domains with neomycin. Okay, so ORF2 has been eliminated right here.
For the neomycin, but
okay, what was the conclusion of this study?
Not exactly conclusion, but we prepared the stable cell line.
Right now we are comparing. One is standard and another one is without ORF1, yes. ORF1, ORF2, ORF1 and 3 are together.
Okay, so the main reason behind preparing stable cell line or the polyethylene of the genome was that just to increase the replication efficiency to study the actual processing of the PCB inside the cell line. So with these two strategies, we used mostly polyethylene for actual or mass spec studies. So that polyethylene, and one was controlled as a GDD mutant, which is just virus, but it will not replicate. And our polyethylene virus will go inside the cell. It will replicate, and inside that, the PCB will show the interactions with the host proteins. So in the beginning, what we did, we showed just with the clones outside the cell line. In the end, with the cell line, with total full genome, we have showed that PCB has a role in the actual polyprotein processing also, and also it is interacting with the host proteins.
So what was your conclusion regarding the mass spectra?
Yes, that is the 600 proteins are modulated by the PCB. Okay. So these are the deconjugations. Deconjugations is like desomylation, deesgylation, denidylation, deubiquitinations. Means it is removing the ubiquitins, somoproteins, then nidylation proteins from the host protein, and it is using that deconjugated proteins for its own replication or for its own establishment. Okay, so generally what happens after the infection, cell first it will try to fight with her own proteins as an innate immune response. And on the other side, if it is going out of the control, it will try to induce apoptosis. So in both the cases, virus will remove that tags which will going to induce the apoptosis, either apoptosis or the either innate immune response, which will trigger the innate immune responses. So that part is mainly done by the papain-like cysteine protease.
Where does this ubiquitination, simulation, and all other modifications occur on endoplasmic reticulum?
Yes, on the
Golgi complex.
the post-translational modification will be means it will continuous from the Golgi apparatus. It will start, but the thing is like even replication will be what the protein synthesis of the viruses will be on the endoplasmic reticulum or nearby there.
Rough endoplasmic reticulum.
Rough endoplasmic reticulum.
Okay, so it is kind of continuous process. So protein synthesis is there, then again further modification is there.
Yes, further modifications will happen, but simultaneously, because tagging with the ubiquitin or ubiquitin-like proteins is associated with the cell functioning and bifurcating the division of the works or division of the labors. So with the specific tags, it has given specific work for the proteins. So what viruses do generally, they remove those tags and they use or they create the confusion inside the cell lines. And those proteins which are important for their replication, they will use. And those proteins which will going to induce the apoptosis and other things, that they will remove from the means they will create the confusion inside the cell line.
Okay. So what is the end conclusion? What happens once this kind of infection occurs? Cell burst or virus remains there, replicate inside, or there is generation of different copies of viral genome or viral capsid kind of whole architecture of virus?
Generally with the RNA viruses, cell actually don't burst like lytic and lysogenic cycles, but continuous aggression will cause the cell death only, but not the bursting of the cells.
Cell death is there.
Cell death will be there eventually. And with the means same again, DNA viruses will have the different strategies. Like when there is actually immunocompromised body, then they will again replicate, and in that case there might be chances of lytic lysogenic cycles, or in case of retroviruses, they are already integrated inside the genome. So in that case there are chances of, you can say, lytic lysogenic, or in case of even bacteriophages, the lytic lysogenic pathways can occur.
Do you think this is a kind of mutual, I don't know what to say, but from virus side, it is trying to make a mutual connection, or from an animal point of view or animal cell point of view, it is not allowing virus to stay inside the cell?
Yes. Any kind of invasion will be kind of hazardous to the cell line. So always cell will rebel or try to create the defense against.
And every time virus will be part of evolution process, adaptability will be there, rate of adaptation will be changed.
Viruses will prefer the adaptation. They will try to remain mild for the
This
cell.
is a never-ending war.
It is never-ending war. So continuously there will be combat between the host system.
So I think at this moment, having memory against a particular bacterial virus, it is not sufficient for human to be in a survival mode or to win the survival.
Yes.
Okay. Your whole study is onto the genome. Okay. You haven't dealt with the original virus structure. Considering all the process that is happening inside the cell, inside the bacterial cell, you made that cell compatible with the viral genome. In case of bacteria, you did same. What do you think about the viral genome? A very few nucleotides fighting against a billions of nucleotides in case of animal cells, and kind of somewhere trying to win the war. Okay. and not allowing cell to die. Again, a second decision is there where it is trying to make a home. Do you think that having no outer structure, just a genome, deciding to stay inside an animal cell or creating a home inside the animal cell and taking some decisions like you mentioned in the post translation modification, removing tags. So somewhere a consciousness is coming here.
Yes.
Do you think that viral genome has some kind of consciousness because of the increase in the rate of evolution?
Yes. So now this is my own opinion. So you can say I don't have experimental proof for this. But overall, when I studied for almost 11, 12 years about the viruses, what I feel, they don't want to kill the cell. Even one more step, they will try to integrate inside the genome, so that now there is no need to even try to combat or try to find out or establish itself inside the cell line. So it will be more evolutionarized that they will integrate inside the genome. So on the both side, whenever I think about the genome and as well as the viruses, in both the sense, I think genome has the consciousness as well as virus genome has the consciousness. Why the human genome or the animal genome has the consciousness? You might have heard about the CRISPR-Cas technology. So it is actually derived from the bacteria. So what bacteria do? Once they get infected by something, they will chop their genome, and for that genome they will integrate inside their own genome, so that it will be act as a memory for so throughout the human genome evolutions, almost we have just 3% DNA which will code for the human proteins. Other 97 is the actual residual impact what has come across the evolution. So almost there are 7 to 8% is the retrotransposons, which I said from the retroviruses.
Selfie DNA.
Kind of. So more than that, there are DNA transposons. So almost 97 genome is not ours. It has come through the evolution. So what my thinking is like, what we are thinking as a individual entity, I am not that person. I am considering myself as a Swapnil Desai, but I am not, because there are more viral genome inside me than my own genome. There are more microbial flora than my own body cells. So we are not what we feel like as an individual human entity. So somewhere I feel like genome is playing with us. Genome is creating combinations and try to find out which will survive the best.
Our body has become home for many microorganisms like salmonella. It is in some organ, like hepatitis E virus. Again, it kind of infects to hepatic cells primarily. Like influenza virus, again, it is having a location. So do you think human body has become a kind of place where everybody is taking home for rent?
Yes.
Or we can say it is a museum.
It is a museum. It is a dustbin of the everything which you want to try, put it inside the human genome and check what will happen. And
it is a silent game that they are playing.
It is silent game. It is. That's why I always feel like, see, suppose we are standing on the top of the hill, and suppose one truck is coming through the guard section. Generally, we know the driver is doing that, moving the truck, but the person who hasn't seen the truck, or the person who doesn't know the driver rides the truck, what that person will think, actual truck is moving, right? But the inside person, he will not that much mature to understand the driver is driving the truck and not the truck by coming itself. So similarly, I feel we are like truck. We feel we are driving our own body and other things, but actual genome and that microbial flora, those are driving us. So there is one more concept. There are few parasites which intentionally try to kill you. Means I don't remember exactly the name of that parasite, but people with those parasites have more prone for the accidents. And in case of mice and cat, there are actual reports. So some parasites have life cycle, half of the life cycle into the mice and half of the life cycle inside the cat. So parasites infect mice infected with the parasite, they will try to go near the cat. So they will try to find the place where mice has urinated. So there will be the probability or chances that cat will again come there and they will be eaten by the cat, and the half of the life cycle will get complete inside the cats. So it is a kind of it is going on different level of consciousness.
What do you think about, I mean, you talked about evolution, right? So what do you think about quasi-gene theory? I mean, there's one quasi-gene, or you can say quasi-species theory. So when virus infects some host, when it goes inside, it has a cloud of its viral genome, like with some related species. So do you think, what do you think about it? Like, is the viral revolution dominated by master gene theory or quasi-species? What's your opinion about this?
Quasi species is multiple mutants. Means obviously when we think about the RDRP, it doesn't have the proofreading activity, so it will make the random mistakes. So thousands of genome might be there inside the single cell, but which one will be coming outside, it is not in. I don't know. Means we haven't reached up to there. So who is deciding what will go outside is not yet known.
Is it a random chance?
It is a random chance, kind of.
Random selection maybe?
A random selection maybe. But again, once the genome size increases, in case of now, we'll talk about the Coronaviridae family, they have the proofreading activity. They have the RDRP with proofreading activity. Why? Because coronaviruses are almost 32 kb, 28 to 32 kb. So for the evolution, again, what viruses have done, if they are increasing the size of the genome, they are trying to remain, they are trying to conserve as much as they can. So mutations are not affordable for them. But with the 8 kb, 12 kb genes, they are trying random compositions to interact with the host.
So it's like they are saving their part for future maybe progenies like that, like just like microform spores.
Yes, they as a genome, they will always carry some kind of information. But again, they are keeping some part. As I said, within the hepatitis E virus also, that macrodomain proline-rich region, they are kind of shock absorber. One more interesting thing is there. There was a patient infected with the HIV and HEV. So ribosomal 40S ribosomal protein is there. So almost 174 nucleotides part of actual human ribosomal protein get integrated inside the hepatitis E virus proline-rich region, and it is kind of safely replicating. So these changes are coming. I don't know whether it is said it is to say randomly, but they are taking chances. They are trying with the different mutational perspectives, I can say.
The chance of propagation after certain, we can say, mistakes, maybe higher or lower. It is random, or it is always higher.
Currently, as per the evolution, since RNA viruses are, since they have come very long, they have traveled a very long journey of evolution. So I think the random mutations are helping them.
So, currently people think that, in the near future, like dystopian future, maybe you can say, like there will be some time where AI will take over I mean, there are different minded people. Some people think that it might take or it might not take, right? So in some of the movies, they show that viruses actually they infect the whole let's say species, human species, and they dominate over there. So do you think that in near future that might be the possibility in upcoming time? Do you think that viruses will take over human genome, or maybe let's say they will fight against humans and they will maybe let's say they will dominate the world? What do you think about this concept?
Are you talking about zombie virus?
Maybe any kind of virus.
So what I feel like, anyhow, always evolutions do the mass extinction. So again, it will do the mass extinction. But what will be the cause for it? Don't know. Maybe viruses or may not be
In
viruses.
the context of animal cell versus virus cell.
Generally, see, viruses. Why I feel like viruses are more evolutionary at more higher version, because as we have as humans, we are the complex bodies. So whenever there is a chances of eradication and extinction, humans will be the first.
Not much adaptable
Not much adaptable.
than that of the virus.
But at a point, again, we are having memories, we are having antibodies. So survival rate is there kind of system. Even we are having intelligence, so we can create antibodies or vaccines. So that aspect is also there.
No, but we are as a human body, we are not taking care of our body. See, like means suppose 2000, 3000 years before, we are totally behaving like animals. So our total focus was on the body. So currently,
Speed of evolution is much more higher in the virus than that of the human cell or animal cell.
yes,
That's the create a very much difference when we consider the adaptive predictability of the virus.
yes. So always chances of extinction of the humans will be more.
Due to the lower rate of evolution.
Yes, lower rate of evolution.
Because humans are high cellular animals. I mean, multicellular animals, high complex animals compared to viruses. So they have, virus can easily replicate in comparison with humans.
So viruses can infect any living organism. So they will remain. Even if there is not the host, human as a host, they have already bacteria as a host, insect as a host, plants as a host.
Everything.
So, they have everything.
So every living thing they can infect, right? And what about bacteriophages? Like they infect viruses, right?
No, bacteriophages are
Bacteria infecting viruses.
the viruses which infect the bacteria are the bacteriophages. Actually, specific DNA viruses.
Not RNA virus.
RNA. Bacteria also can be infected by the RNA viruses, but the bacteriophage specific term is used for the DNA viruses which infects the bacteria.
Bacteriophages.
So can you just give us brief overview about bacteriophages, like how they act on viruses, how they basically insert their genome inside virus? I mean, how does it work in case of bacteriophages?
What you have studied in your academics, that is the actual example of bacteriophages infecting the cell. So they will, you might have seen like
Phages basically.
So it is the common thing. But again, the interesting part is even nucleus might have come from the viruses. There are the chances. Why? Because when even DNA viruses first infect the cell, they try to create the nucleus-like region, and inside that they will replicate. Even in case of hepatitis C viruses, they try to create their own environment for the replications. So it may consider as pseudo-nucleus kind of things.
So you mean bacteriophages, right?
Bacteriophages also do. They means generally DNA viruses. Not all the DNA viruses will go inside the genome. So some of them will remain inside the cytoplasm, but they will create their own nucleus-like structure
Okay.
and they will replicate there.
Okay.
Let's end.
Okay.